Bismillahirrahmanirrahim. Assalamu alaikum, dear student. My name is Dr. Muhammad Adnan Asgar, and today we are talking about characterization of pharmaceutical materials by using thermal techniques. Today is actually the last lecture on the pharmaceutical materials. Uh, Here is protein denaturation. Protein denaturation is a general term used to describe a change in structure of a protein. This change usually results in a non-reversible unfolding of the protein that affects its shape and therefore its biological activity. There are variety of techniques that is used to measure protein denaturation, including change in physical properties that may be the solubility and change in reactivity such as with enzymatic protein. So these are the two important factors. With diffraction scanning calorimetry, protein denaturation is actually the measurement of the thermal stability of a protein in a solution. A low energy endothermic peak is observed over a temperature range. The temperature of the peak actually provides information about thermal stability, and the area of the peak is actually a quantitative measure of the energy absorbed by the protein in order to change structure. Experimental conditions such as heating rate, pH, ionic strength, that is salt concentration, and even protein concentration can affect results. In order to minimize the opportunity for aggregation of protein, most measurements are performed at relatively low concentration. A relatively low concentration means 1% or 10 mg into 1 ml of the solution. Diffraction scanning calorimetric data for the denaturation of albumin from chicken eggs is shown in this diagram, which is actually the comparison of 1 and 10% concentrations. So this is the diffraction scanning calorimetric figure, and it is for the actually albumin which is extracted from chicken eggs, and it is the comparison of 1 and 10% concentration. At 1% concentration, a peak of at 1% concentration, a peak of about 40 microvolt in height and 0.21 joule per gram in area is easily detected. The sharpness of minor detail associated with peak is however less clear because of low signal strength. At 10% concentration, the peak is about 400 microwatt in size, and there is excellent detail showing minor shoulders on both the low and high temperature sides of the peak. The fact that peak temperature differ by only 0.2 degrees Celsius provides confidence that minimal aggregation occur at 10% concentration. So there is a great flexibility in analyzing data. In this figure, the 10% concentration data is shown with the sigmoidal baseline, and percent denaturation is plotted as a function of temperature. A plot of time during the experiment shows that the entire experiment took only 60 minutes, which is faster than typically obtained with microcalorimeters or solution calorimeters. So this is actually the elaboration of protein denaturation, and we can analyze the different concentration and by using diffraction scanning calorimetric technique. And next one is the freeze drying. Freeze drying or lyophilization has become a standard process in the pharmaceutical industry for the manufacture of biologically active substances. However, it is not without limitations due to its high cost in capital and energy, and we can said even in long processing time. Moreover, the difficulty in selecting manufacturing conditions of time, temperature, vacuum, and component concentration. All of these parameters must be optimized in order to achieve a final product with the desirable characteristics of full activity of the protein or drug, easy reconstruction, acceptable appearance of freeze dried cake, good storage stability are actually the parameters that must be optimized in order to achieve a final product with these desirable characteristics. Moreover, the process of freeze drying relies on the vapor pressure of ice. Even at temperatures as low as minus 50 degrees Celsius, ice sublimes and leaves a very porous, low density cake containing the stabilized drug. Since the sublimation rate is very temperature dependent, use of highest possible temperature during primary drying actually provides maximum drying efficiency and low process cost. In order to select the optimum drying temperature, it is necessary to understand the physical characteristics of components used in the formulation that is to be freeze dried. In decreasing order of mass, these are typically water, bulking agents, buffers, or stabilizers, and finally the drug itself. The bulking agent, which can either be crystalline or amorphous in nature, and its interaction with frozen and unfrozen water in the frozen solution actually define the physical structure which is essential to the successful freeze drying. This structure manifests itself in the form of transition that occur at specific temperatures. Physical properties of the bulking agent, such as modulus or viscosity, can change by orders of magnitude depending on the process temperature is a few degree above or below the transition temperature. Therefore, the knowledge of this structure and how it changes with time and temperature as well is required for the successful drying. Diffraction scanning calorimetry has been used with only modest success in the characterization of frozen solutions used for freeze drying. The reason is that there are numerous components and transitions happening within a narrow temperature range, and diffraction scanning calorimetry can only measure the sum of these. In addition, diffraction scanning calorimetry must use relatively high heating rates. These heating rates may be 10 to 20 degrees Celsius per minute in order to optimize sensitivity, while the best resolution is obtained at low heating rates of zero point 0.5 to 1 degree Celsius per minute. In the introduction section on new diffraction scanning calorimetric technologies, it was explained how modulated diffraction scanning calorimetry has both an average and modulated heating rate. The combination of two heating rates allows the operator allows the operator to select a slow average heating rate in order to obtain good resolution, and a higher modulated heating rate to obtain increased sensitivity during the same experiment. In addition, the resulting modulated heat flow can be separated into the heat capacity and kinetic components of the total heat flow in order to improve ease of data interpretation. This in this figure actually shows the raw modulated heating and modulated heat flow signals from a modulated diffraction scanning calorimetry experiment. which is performed at an average heating rate of 0. 0.5 degrees Celsius per minute on a frozen solution of 40% sucrose in water. The change in heating rate, which is actually the modulated one, is approximately 3 degrees Celsius per minute, which causes the modulation in the heat flow signal. In this diagram, it the calculated signals from this modulated diffraction scanning calorimetry experiment. The total signal is very difficult to interpret because it is equivalent to standard diffraction scanning calorimetry at the same heating rate and contains two different transitions. The first of these is the important glass transition seen in the reversing signal between minus 43.6, here we can see, and minus 39.41 degrees Celsius. The second transition is an exothermic in the non-reversing signal, which is actually caused by crystallization of free water that would not crystallize during the quench cooling of the sample. This peak shows a maximum at about minus 36 degrees Celsius and a heat of crystallization of 5.7 joule per gram. Here we can observe the values. It is not surprising that unfrozen water would begin to crystallize near minus 42 degrees Celsius, since this is near the onset of glass transition where a significant increase in molecular mobility and diffusion can occur. In this modulated diffraction scanning calorimetric measurement, we have observed the two types of peaks, and while the first of these was the actually important glass transition, which is seen in the reversing signal between minus 43.66 degrees Celsius and at minus 39.41 degrees Celsius. The second one is an exothermic in the non-reversing signal caused by crystallization of free water that could not crystallize during the quench of sample. This P shows a maximum at about minus 36 degrees Celsius and heat of crystallization would be the 5.7 joule per gram, and it is quite obvious in this diagram. So there are two type of transitions can be observed, which is well captured by the modulated diffraction scanning calorimetry. In this diagram, uh, even modulated diffraction scanning calorimetric data shows the obtained which is obtained on the same sample, except that it was cooled at zero point 0.5 degrees Celsius per minute as compared to the quench cooling system that was used in the last one. The cooling and heating data are actually plotted in heat capacity units so that they can be visually compared. Notice that the slow cooling produce a more complex structure which has two step changes in heat capacity. The derivative signal more clearly shows that the double transition, and it also shows that there are minor temperature lags between heating and cooling with T0 diffraction scanning calorimetric technology. Even at slow average heating rate of zero point 0.5 degrees Celsius per minute, modulated diffraction scanning calorimetry provides extremely high sensitivity for the characterizing of the complex structure of frozen solutions used for freeze drying. An modulated diffraction scanning calorimetric experiment is uh, mm, if we performed experiment on the same instrument as used for the traditional diffraction scanning calorimetric measurements. The difference between two techniques is that the temperature profile applied to the sample and the deconvolution, which is actually the separation of the resulting heat flow signal into several components. Instead of simple linear temperature change used by diffraction scanning calorimetry, modulated diffraction scanning calorimetry uses two simultaneously heating rates. An average or underlying the rate which is similar to the diffraction scanning calorimetry plus a sinusoidal or modulated heating rate. The average rate provides information equivalent to traditional DSC, while modulated heating rate provides unique information about the sample heat capacity. Moreover, traditional diffraction scanning calorimetry provides a single signal which is the sum of all thermal events occurring within a temperature range of the experiment. This often makes it difficult to interpret data or detect small transitions. While modulated diffraction scanning calorimetry has a significant advantage over the traditional DSC in that it measures both heat capacity component and the total and obtained kinetic component from differences. In general, modulated diffraction scanning calorimetry provides the following advantages over traditional diffraction scanning calorimetry. It has increased sensitivity. The modulated diffraction scanning calorimetry actually possesses increased sensitivity, increased resolution, separation of complex transitions, more accurate measurement of crystallinity in semi-crystalline materials, direct measurement of heat capacity while programming temperature or holding it isothermal. So these are the some of the advantages of modulated diffraction scanning calorimetry over conventional diffraction scanning calorimetry. In this experiment, we can observe that modulated diffraction scanning calorimetry successfully separate the complex transitions in a compound. So it have ability to separate the complex transitions with more sensitivity and high resolution. This is all about this uh, lecture, and here are the some references. We need to learn this kind of lecture from the book, which is the Thermal Analysis Fundamentals and Applications to Material Characterization by Dias, and one of the research article entitled as Characterization of Pharmaceutical Materials by Thermal Analysis by Thomas. Thank you very much.